Welcome back to our visual presentation dealing with the research topic and the research title, in which now we will try to discuss how to identify the research problem in line with your research topic for your SIPs. So note class that the major aim of research is to solve the existing problems in our community so that we can achieve a comfortable life. Therefore, we can define research problem as any situation that is perceived to be challenging and needs improvement, changes, and solution. Okay, so with this definition class, we can say that if we have a research problem, it means that there is a need for us to conduct research. Okay, but how do we identify a research problem for SIPs? So let us have the following guidelines and examples. So note class, in your SIPs, before you could formulate your research title, you must first choose a category where you wish to participate in. And we have four SIP categories. We have life sciences, applied sciences, robotics, and research innovative. So, for our first guideline in identifying a research problem, you must first align your research problem to be investigated into different categories. Okay? So, you must choose your category of interest. Remember class that if you choose an SIP category, it must trigger your curiosity. Okay, so that you can show interest, have more motivation, and more enthusiasm in conducting your study. So we have here the four categories of SIP. We have life sciences, applied sciences, robotics, and research innovative. So class, for each category, there are actually still different, let's say, disciplines, subfields, and subcategories. Okay? So let us try to identify what are the possible subcategories for each SIP category. So for life sciences, okay, we have the following specific fields. So your study under life sciences can fall under the discipline of animal sciences or zoology, cellular biology or cytology, medicine and health sciences or medicology, microbiology, and plant sciences. Okay? For applied sciences, your study could fall under the disciplines of chemistry, computer sciences, earth and planetary sciences, engineering, environmental sciences, physics and astronomy, and material sciences. Okay? For robotics, actually robotics class, it coincides with applied sciences. However, the difference is that in robotics, you must have an let's say a main output that is actually a robot. Okay, and this robot is will serve as the product of your study, and it will encompass the topics under computer programming. Okay, so that is, or let's say that is the subcategory for robotics. Now for research innovative, okay, so research innovative category is somewhat, let's say, a combination of the three other categories. However, the main distinction of research innovative category is that you will be required to innovate, or let's say upgrade an existing product. Okay? You must upgrade an existing product for its further development. Preferably, it will add technological advancements, okay, to the existing product you wish to innovate. Okay, so these are the different, let's say, differences of the SIP categories that we have. Therefore, our second guideline for you to follow in choosing a research problem is to dissect the categories of SIP and then choose and focus on one subcategory of interest. Okay, choose a subfield. Okay, so after choosing a subfield, what is our next step? So let's try to analyze the next situation. So we have here, okay, let's say we have chosen life sciences as our SIP category of interest. Now, after thorough consideration, let's say you identified that you wanted to focus on the study of microorganisms known as microbiology. However, microbiology class is still a broad discipline. So, we need a more specific area. So with that, you decided to focus on the study of bacteria, known as bacteriology. However, bacteriology can still be a bit vague. Okay, it can still be a bit broad. Therefore, you decided to narrow it down further into the study of human bacterial pathogens, known as human bacterial pathology. Okay? So, bacterial pathogens are those bacteria that can cause harm to humans. Okay? And from this part, okay, from this dissection of such topics, now you would want to identify a certain, let's say, variable, okay, or experimental unit for you to choose, or let's say for you to utilize in your study. Now, this variable that you would want to consider or you would want to, let's say, accumulate can actually be a potential source of problem in the field you have chosen. Okay? Again, after choosing or let's say narrowing down your subfield, you must choose a variable that can cause a potential problem in the discipline you have chosen. Okay? And upon further research, you found out that a bacteria known as Salmonella typhi, okay, causes a wide array of problems in the microbiological field. Okay? So from here, class, you can observe such pattern from a broader category, which is life sciences. Okay? We try to narrow it down to subcategories and then to other specific areas or subfields, and then we identified a potential variable. Therefore, class, our third guideline in identifying our research problem is for you to identify a potential variable, experimental unit, or situation that causes or might cause problems to the community that is related to your discipline of interest. Okay? And note here, class, that most of the time, okay, the identification of our, let's say, potential variable and experimental unit will actually identify our dependent variable or DV. Okay? So again, the identification of our potential variable, experimental unit, or a situation will lead us to the identification of our dependent variable. And actually, class, the identification of such dependent variable will lead us to our next guideline. So let's take a look at our next example. So after identifying Salmonella typhi as our, let's say, potential variable that can cause harm, you found out that in a certain community, there is a huge outbreak of a disease known as typhoid fever. Okay? And this typhoid fever, class, is actually a disease caused by Salmonella typhi. Okay? So typhoid fever can be acquired upon the consumption of contaminated water. Okay? Note, class, that Salmonella typhi is a waterborne bacteria. So when we say waterborne, it spawns or it lives in water environment or aqueous environment. Therefore, class, typhoid fever is a waterborne disease, okay, which we can acquire through the consumption of contaminated water. Okay? So, how can we categorize the occurrence of typhoid fever? Okay, class. So the outbreak of typhoid fever caused by Salmonella typhi is actually our existing problem. And that will be our next guideline. So, you must identify your rational, okay? And your rational class is actually our existing problem. And actually, our existing problem is also our research problem. Okay? So you must take note, class, that the category you have chosen from the very beginning should have a connection to your rational. So notice, class, that from life sciences, we identified our rational, meaning to say there is a connection. Okay? So with these steps, class, in identifying the research problem, what did you notice? Okay? What approach did we use? From category to subfield to variable to rational. Okay? Did we try to use a broad to specific approach? Okay. Obviously, class, the answer is yes. And this kind of approach, wherein we go from broad to specific, will be known as the deductive approach. Okay? So meaning to say, class, we must identify our research problem through the use of deductive approach. However, class, the process does not stop here. Okay? After we have identified our research problem, it means that that will be the time that the researcher must provide solutions to their existing problem. Okay? And of course, we can provide a solution by conducting a study. Okay? And every study has their own research title. So class, meaning to say, after identifying the research problem or the rationale, we are now ready to write our research title. And that will be the main concern of our next video presentation.